Good afternoon, everyone, to the close and to the far people, which actually are 0. But maybe they will increase with time soon. Okay. Good afternoon. So, the title of the presentation more or less says it all. I'm going to talk about why it's important to measure mechanical properties for biological samples. I will show you examples of this at different scales, going from tissue to cell membranes, and I will show you why atomic force microscopy offers a great potential in this field. So, okay, let's first of all let's say what do we mean for mechanobiology. For mechanobiology, we are meant to measure how biological specimen sense and respond to mechanical stimulus from the external environment or from external objects, how can be the probe of a microscope, and how these mechanics influence their overall patho-physiological state. And as I said before, okay, I will show you some few examples of biomechanical measurement by atomic force microscopy on different biological specimens, for example, cells and extracellular matrices on different scales too, for example, cells and tissue mainly on the micro scale. Then I will show you an example about the interaction between cells and engineered by biocompatible surfaces produced in order to mimic the extracellular environment. We will see some features on the micro scale as well on the nanoscale. To end with let's say the topic of interest, more of interest for this project, which is the model of cellular membranes and their interaction with extracellular vesicles, and this will be focused, of course, on the nanoscale since it is definitely a nanosystem. So it's well known since decades that the mechanical properties of biological specimens are very important. For example, the cytoskeleton of cells, which is one of the main components of cells, besides providing a structural role, that is providing the scaffold for the cells, is also responsible for many primary cells function as adhesion, proliferation, migration, differentiation, and many others. So it's clear that the mechanical phenotype of cells is directly correlated to the cell's vital parameter. At the same time, cells don't live alone. They don't live in a bubble, but they constantly interact with the surrounding environment, which is mainly, first of all, the so-called extracellular matrix. Is it okay? Okay. With the extracellular matrix. No, ho visto solo. Okay. Which is, let's say, an ensemble of biological molecules, proteins surrounding the cells. And from a certain point of view, it provides as the cytoskeleton the scaffold where the cells live, but it's also important for regulation of some vital cells function like migration, growth, evolution, building up of anchorage points. And so as for cells, it's important to study the correlation between cellular behavior and extracellular biomechanical properties. Last but not least, if we go a little bit on a smaller scale, on the nanoscale, and we focus on a small part of the cells, which is their skin, their membrane. Their membrane is a complex system, but is very important because it's the gate. So everything that goes out or in has to fuse in some way with the cellular membrane. And also because the cells communicate with each other, exploiting the so-called extracellular vesicles, which are also built in some cases using the help of the cellular membrane. And of particular interest, they have different dimensions, but of particular interest are the so-called small extracellular vesicle with a diameter around from several tens to a couple of hundred nanometers. How are they made? They have an outer shell which is similar to the cellular membrane, is a complex structure made of lipids, is a lipid bilayer enriched with different proteins, extracellular, extramembrane proteins, transmembrane proteins, ligand receptors, and they also carry some cargo inside, like nucleic acids or several kind of proteins. So from this description should already be clear that they can have a heavy influence on the fate of the recipient cells, depending on the type of cargo they carry inside them. But of course, they have, as I said before, interact with the membrane before releasing, before entering the cell and releasing their cargo. So it's important to study the membrane itself, but also its connection, its relation with extracellular vesicles. There is another reason why they are important. I mean, the small extracellular vesicles. The reason is that they are very similar to some kind of viruses. So they can be considered like virus-like particles. They are similar in dimension, especially, for example, with the SARS-CoV virus. They are similar in some cases in the origin, how they are originated, in terms of also function. They have a cargo that they want to release inside the cells. Of course, the proteins that are connected to the membrane, to the external bilayer are different. The cargo is different. Also, the internal structure of enveloped virus is a little bit different because they have also an internal capsid, but in some aspects they are somehow similar. And it's also possible, this is already an ongoing collaboration with University of Turin and our group. It's also possible to make cell express extracellular vesicles with spike proteins on their outside membrane in order to mimic the viruses, the enveloped viruses of the SARS-CoV, of course. They are not the same, exactly the same things, but if we don't have, biological labs with a certain level of safety, we can try to use mimicking tools like this. So in this context, how can an instrument like atomic force microscope be important. Let's say that nowadays the research is going towards an increasing level of multidisciplinarity and versatility, because many topics like this are covering several disciplines, and the specimens are way more complex and need also versatile tools. And in this kind of context, atomic force microscope fits very well because it provides a wide versatility, starting from, for example, specimen and environmental condition, because it can measure insulator as well conductors, materials, can operate in every kind of environment. It can operate on inorganic core as well organic samples, and it allows to vary on different spatio-temporal scales, as I will show you. On the spatial side, it can go from micro to the nanoscale, or in some specific condition, even atomic resolution. And from this point of view of temporal scale, it can go from minutes to, let's say, microseconds. Minutes are the more conventional, traditional AFM, whereas microseconds are the super expensive high-speed AFM, like the one that we have bought for this project. This is a nice slide. I don't know if it's clearly visible. I think yes, which shows things in biology, what can do IFM in this context. So you can see that conventional image scan AFM are on the time scale of minutes or tens of minutes, whereas with high-speed AFM, we can reach the world of milliseconds, and if you were to reduce the spot analyze also to microseconds, and so we can explore the dynamics of biological macromolecules or proteins, which can be of really high interest. But I will explore this. I will show something more detailed about this later. So I don't know how many of you are familiar with the working principle of atomic force microscope. I will just be quite generic. I've stolen this slide from a colleague of mine, which I really thanks, because it's very nice. I mean, it gives the concept in a really easy, delivers the concept in a really easy way. How does an atomic force microscope basically works? It's like having a nano finger, kind of nano finger, that is moving over a surface and following the topography. Then in practically how it is realized, this nano finger, it's a very sharp tip whose height can be some several microns, also 10, 12 or more microns, but the important part is the apex of the tip. So the atoms at the apex of the tips, which are interacting with the surface of the sample, and they are feeling the force interacting between the samples and the tip. The tip is attached to an elastic lever, which is called the cantilever, and that is a force sensor. And there is a laser which is reflected over the backside of this force sensor or cantilever, then ending on a photodiode, which can easily measure the deflection both on lateral and vertical direction of this lever. So on one side, this led us to follow the topography of the sample on very small scales, but also it's very important, we can directly measure the force acting between the tip and the sample, and this is very important because these force curves, like This example you see on the right hide very important information that can be combined to topography. For example, when the tip is pulled toward the surface in the so-called attractive regime, you can measure the adhesion between the tip and the sample, which can provide you a chemical information on the nanoscale about the property of the surface, so chemical properties of the surface on the nanoscale, whereas when the tip when the cantilever starts being bent upwards, it means you are going in the repulsive regime. You are also squeezing maybe a little bit the samples, and from this part of the force curve, for example, you can get mechanical parameters of the sample under investigation. So you can build several maps in one-to-one correspondence topography with, for example, for our case of interest, elastic parameters. Versatility of AFM is also evident in the fact that you can change the tips according to the type of measurement you want to do. Of course, if you want to measure something on the nanoscale, like I don't know, the roughness of a nanostructured surface, you can use the standard AFM tips, whose final radius of curvature can reach 10 nanometers or even lower with more modern tips. Whereas if you are interested, for example, in the mechanical properties of a cell, but not of a single fiber, let's say on a network of fiber on portion of the cytoskeleton, you can change the tip and replace it with a spherical bigger one with the diameter in the range of, for example, 10 microns. So it depends on what is your purpose, your final goal. You can change the characteristic of the instrument. So let's finish this introduction and finally present some example of application. So about cells elasticity, during my PhD, I've been working first of all on the elaboration of a protocol which could let us to measure at the same time in one-to-one correspondence on cells cultured, so not fixed with any kind of glue, but cultured on their medium culture, so in let's say physiological condition. We could by this protocol obtain combined one-to-one corresponding topographic and mechanical imaging of single cells. What you see here are 3D force maps. I mean the height of the maps is given by the color scale. On the top you see the height, so the topography of the cell is a PC12 cell, whereas on the bottom you see the mechanical parameter, which is the Young's modulus, which is an intrinsic parameter. It means it depends only on the material of the sample, not on geometry or shape or dimension or volume or other extrinsic factor. So I will skip this because I don't want to go too much into technical details, but basically the technique that I use to make this simultaneous measurement is called force volume or force map. Nowadays, with this new high-speed AFM, we can apply the same technique, but going much faster compared to the times I was used to, for example, to acquire the previous images, 100 squared microns and 64 per 64 pixel. It took me around 30 minutes, whereas nowadays we can go much faster in principle. So another example is measurement of mechanical properties, or an example of application of this protocol is the measurement of mechanical properties of extracellular matrices in the framework of a collaboration we did with San Raffaele Hospital in Milan. They provided us with samples from donors related to colorectal carcinoma. They were purified extracellular matrices, so yes, so they had a protocol in order to get rid of cells and only keep the extracellular matrix without affecting the original properties of the matrix. So they provided us with, let's say, pieces of biopsies from the tumoral regions, the perilesional, so the region surrounding the tumoral region, and the healthy regions, let's say, around 10 centimeters far from the tumor region. We measure the mechanical properties through AFM of the samples. On the left side, you see only the statistics of a single donor, whereas on the right side, you see the statistics of 3 donors and the overall statistics of the donors put all together. And what we observed is a gradual increase of stiffness of the ACM going from healthy to tumoral region. But AFM alone cannot provide every useful information to get a comprehensive and detailed picture of the problem under investigation. As I said before, we are, especially if we are going to investigate multidisciplinary topics overlapping in several fields, and so AFM could provide us information about mechanics, but for example, scanning electron microscopy could provide us information about the topography of the samples, which was showing a gradual increase of linearization and coalescence of the fibres composing the CM, and some other experiment performed by scientists at San Raffaele Hospital showed us that in reality, healthy and perilesional ECMs share the same protein composition, so there is no difference in the protein composition of healthy and perilesional, but there is an increase in collagen cross-linking. So this is the key in moving an healthy ECM towards a tumoral ECM, and this is done in order to also to favor the vascularization of the region, which is indeed needed by tumoral cells. And so this correlates this progressive linearization and coalescence of fibers correlates very well with the increase of stiffness measured by AFM. And so why it's important in this case to get this measurement? First of all, you can think about using measuring ACM properties as a screening. So the stiffness of increasing stiffness and cross-linking of, for example, ECM coming from some biopsies could suggest potentially the starting of a neoplastic region, and that also can be used as for therapy purpose if we imagine not to target the cells but the environment in which the cells live, since it has been proved that tumoral cells seeded into healthy tissues do not proliferate. So there is a mutual interactions, cancer cell needed a proper environment. So targeting the surrounding SEM, like with antioxidant agents, antifibrotic agents, or trying to normalize the vascularity of the region can help as therapy tactic, let's say. Then we'll try to get faster. We can also study the interaction between cells and their extracellular environment in a different way. For example, we tried to mimic the roughness of the natural roughness of extracellular matrix with engineered biocompatible nanostructural surfaces. We studied with sharp tips, so we studied the average properties of these nanostructured surfaces, such as roughness, and with larger micrometric tips, we measured the elasticity of the cell on the micro scale. And what we observed is that a certain degree of roughness is able to induce the differentiation of cells without the help of other chemical factors, like in this case nerve growth factors. And going more in detail, for example, this is a very nice cryo-TEM image. We could study the interaction site of the cell surface, the cell membrane, and the underlying nanorough structures to see that actually the cell was not following the entire profile, peaks and valley of the nanostructured surfaces, but was focusing mainly on some peaks depending on their area, their aspect ratio, and it was providing some spatial constraints which were then telling the cells from mechanotransduction paths if to grow classical focal adhesion or different kind of adhesion, so at the end influencing the final state of cells. So coming to biological macromolecules, so the topic of interest for our project nowadays, cell membranes are quite complex objects. They are made of many kind of lipids from different families and proteins, ligand, receptor, whatever. What we can do is trying to model again these cell membranes. Unfortunately, we cannot take the skin directly from the cells and place it on a substrate, but we can try to model these membranes by mixing several lipids and especially cholesterol. Cholesterol is recognized to have a very important role in modifying the membrane fluidity and also their phase equilibrium. Indeed, in this case, sphingomyelin usually is found in a gel state, let's say more solid ordered state, but with the presence of cholesterol, it induces a less ordered state. So what we see here is a map of the topography of a cellular model membrane with some small islands made principally of sphingomyelin and cholesterol, but we don't speak about gel state. We speak about liquid ordered for this island, liquid ordered state, and liquid disordered state for the background, let's say. Then our group has already done a lot of works about this system, especially studying the interaction between the membrane and several kind of small extracellular vesicles, studying the fusion process, so the way they interact with the model membrane, and they try to release the cargo eventually inside the cell. And what they have noticed is that also extracellular vesicle coming from different cell lines behave in a different way, also because they have a different membrane composition and interact, of course, in a different way. For example, vesicle coming from tumoral cell lines like MDA, MB231 cell lines try fuse directly with this small raft island, and progressively destabilize this island, and also induce the formation of pores, these black kind of areas that you see in the image, which are possible, let's say, path for the release of molecular cargo of the vesicles, whereas vesicle coming from non-cellular types behave in a different way. They preferentially attach to the sides of this island, and they make higher patches protruding for a nanometer above the surface of the bilayer, and they progressively increase the area by recruiting material, let's say lipids, coming from other of these small islands with time. But of course, you can see more details in the references that you see cited in these slides. And it has been also studied that they, of course, can affect the fate, as I was introducing before, the fate of the cells. Our group has been performing a mechanical measurement on cells which were receiving extracellular vesicles by breast cancer cells. And what they observed is that this was inducing a mechanical change mainly in the deeper part of the cells. So that one related more to the nucleus of the cells, and this was further proved by another technique here, which told us that actually we have a kind of decondensation of chromatin inside the nucleus, and this explains for change in shape of the nuclei, and also a decrease in the stiffness of the nuclear part of the cells. So I'm coming to an end. I know I'm late, but just to tell a few things about membranes nanomechanics, which should be, let's say, my main focus on this project. Of course, this is still an ongoing work, because I faced several issues, because membranes are complex systems. Cellular membranes are complex systems. There are problems more complex, let's say, compared to cells on a wall seen from the micro scale point of view. So we have several problems, for example, in theoretical modeling and data analysis, since some scientists are not sure if to use, if they behave in a fluid way, or they behave more in a solid viscoelastic wave, so the models change. If we like a fluid-like response, we have to use, let's say, Dusk-Connelly model, which are quite complex equation, and also they are available in an analytical form only for some lipidic composition, or for example, for others, for example, the ones involving sphingomyelin, No, we cannot obtain an analytic form, and we should be forced to solve numerically quite complex integral equations. Whereas on the other side, if we use the more common Earth models typically used for cells and tissue, on the micro scale, it's one matter. On the nanoscale, it's another matter because, first of all, it's more accurate, let's say for absolute values for spherical micromagnetic probes, is less accurate if we take into consideration the shape of nanometric AFM probes, and is more suitable, of course, for the fitting of the initial, because it assumes the samples to be viscoelastic. So it is more suitable for the fitting of the initial compression, initial deformation close to the contact point, but sometimes the precise localization of contact point, that means when the probes getting to real contact with the surface, is not straightforward and may be hindered by false contact region, like due to the presence of long-range forces like electrostatic interaction or other kind of forces. And on the side of experimental part, we have also complication. What we would like to measure is a curve like this, where you see flat region when the probe is not in contact with the sample, then an increase in the force applied to the tip when you deform it slightly, then a sudden kind of drop when you go into breaking into the rupture of the membranes, and then straight vertical line when you get in contact with the substrate. You can get information by analyzing, let's say, the region comprised by the point B and C, but also the force related to the breakthrough, because more stiff samples are likely to have to show higher breakthrough forces. But this is what we would like to see, what would be nice to obtain. A topographic map in one-to-one correspondence with a map of breakthrough force and Young modulus. Even though this is a paper from 2010, but I have some, let's say, some doubts on the way they obtained these results, because the samples basically look like frozen, whereas we know that dynamics of this island on model cell membranes are definitely faster. But what we usually see at the moment, we see two breakthrough events, which are likely due to the formation of a bilayer on the tip as well, and this leads to very ambiguous data interpretation. So we are trying to face this problem by getting back to two strategies, possibly getting back to old-fashioned silicon nitride tips, which are not so nowadays they are not widely used because they have a higher, let's say, lower resolution, or to develop a protocol for the tips for functionalization in order to make them less hydrophilic and avoid the possibility of formation of this bilayer also on the probes, which makes actually impossible to understand from the first curve which part we have to analyze, or if we are measuring the properties of the bilayer attached to the probes instead of the one deposited on the substrate. So we skip conclusion and perspective. What I was just to highlight is that, of course, collaboration is very important in this kind of research, collaboration between scientists and from different fields. And as I have shown you in the example before, also different tools and instrument in order to have a complete and detailed pictures of the problems. And with this new instrument that we recently bought, we can also, we hope to be able to get insights on protein and single molecule dynamics, like you can see an example in this previous last slide. These are examples of what high speed AFM can do in biology. That means basically filming the temporal evolution, the temporal dynamics of proteins, or also part of enzymes, or as you can see on the right part of the slides, the dynamics of surface spikes, which may play an important role, of course, in the interaction between virus and membranes. Okay, I've been quite long, so sorry for that, but I finish.